Good evening, everybody, and welcome to chapter 4, and that will be genetics and cellular function. So, in the past, we talked about the main macromolecules. We talked about carbohydrates and their building blocks, those little glucoses, fructose, galactose coming together to give us lactose, the big one, the disaccharide, and disaccharides giving us carbohydrates in the form of glycogen, in the form of cellulose, and so on. So that was one category. The carbohydrates is a macromolecule. Then we talked about lipids. That's a macromolecule. Then we talked about proteins made up of amino acids. Lipids are made up of what are lipids made up of? Triglycerides. And lastly, nucleic acids. Nucleic acids are made up of nucleotides. So today we're going to talk about the DNA. What is this DNA and its importance? And the most important thing to know about the DNA is the central dogma of biology. The central dogma of biology. What is the central dogma of biology? The central dogma of biology says that the flow of genetic information, is unidirectional. So you begin with DNA, that's the central template, and you transcribe the DNA, you do transcription of the DNA inside the nucleus, and then you get mRNA, messenger RNA. There's different kinds of RNA. We will learn today. Messenger RNA, mRNA, transfer RNA, tRNA, ribosomal RNA, or RNA. But anyways, you gotta transcribe DNA to get RNA. And from RNA, you're gonna get proteins if you successfully translate. So you perform translation of the RNA in the ribosomes. And we have two kinds of ribosomes. We have free-floating free-floating ribosomes in the cytoplasm, and we have endoplasmic reticulum bound ribosomes. These are bound to the endoplasmic reticulum. But that's the law. You go one from DNA, you transcribe DNA, you get 2 RNA, then finally you get 3 proteins. The proteins are going to be either structural proteins serving structural integrity, and then you have functional proteins. So you have structural proteins and you have functional protein. That's the central dogma of biology. So you're never going to get RNA unless you transcribe DNA. And you're never going to get proteins unless you translate the RNA. So now you have to, because we already talked about the cell. We divide the cell into 3 compartments. The plasma membrane, that's the outer compartment. Then the cytoplasm, that's the middle compartment. It has, remember we said the swimming pool and the rubber duckies. And we said the swimming pool is the cytoplasm, is a cowboy, and the rubber duckies, these are your organelles. And then you have your nucleus that has the DNA inside it. So now we're inside the nucleus. Inside the nucleus, you have the DNA. And the DNA is made up of long strands known as chromosomes. You have multiple chromosomes. Each chromosome, if you take it out, each chromosome has a gene. Each chromosome has a particular gene on it. So this region is gene 1. This is gene 1. This is gene 2. This is gene 3. This is gene 2. This is gene 3. So let's say you want a particular protein, like hemoglobin. Let's say you want hemoglobin. So you gotta go in the DNA for the hemoglobin gene. This is hemoglobin gene. This is albumin gene. This is glucose transporter gene. These are all different genes. If I want hemoglobin, which gene am I going to go to? Gene 2. So when you go to gene 2, what are you going to do to it? You're going to transcribe gene 2, and then you're going to get mRNA of gene 2. Then you're going to translate gene 2 mRNA to get your hemoglobin. So each region is particularly specific coding for a certain protein. No gene is going to code for 2 different proteins. So now that brings us to an important concept. When you look at the cells under the microscope, like we did last week, the bone cell look different from the muscle cell, looks different from the nerve cell, looks different from the red blood cell, looks different from the fat tissue cell. Why they all look different, even they all have the same DNA, right? The DNA is the same. The DNA of all organisms within the same species, obviously the DNA in my nucleus is different than the DNA in the frog nucleus, is different from the DNA in the horse nucleus, is different than the DNA of the dolphin nucleus, because we are different species. But if you are the same species, all frog, all human, all cows, is your DNA going to be different? No, your DNA, if you are in the same species, if you are all frog, if you look at all the DNA, is it going to be different? No, the DNA is the same as long as you are in the same species. But that's interesting. If you look at species, so species meaning, so species, that is, are you frog? Are you human? Are you an elephant? Are you an elephant? And so on. Okay? So if you are the same species, your DNA is the same. So now we are human species. But does your bone cell look like your nerve cell? Does your nerve cell look like your muscle cell? No. So how come? Because it's a different gene. Yeah, you're very true. Because the DNA is the same. The DNA is the same. But the active part. So if you look at a human, all human nerve cell, human liver cell. We call it hepatocyte. Human nerve cell, we call it what? Neuron. Human muscle cell, we call it a myofibril. And we have human bone cell, we call it osteocyte. If you look at all of them under the microscope, they don't look the same, even though they all have the same DNA. So the reason is it has to do with the genes. Because for the osteocyte, the bone, the gene that will give me proteins that will help the bone structure are active, while everybody else is off. Everything else is off, and this one is on. You move to the other one, muscle. The muscle gene, the muscle proteins are going to be on, and the bone that was off that was on is going to now be off. It's going to be off. Everything is going to be off. So that shows you, even though the whole DNA is there, but only certain region that give the cell what it needs structurally and functionally. That's what helps. That's what's active. That is what is active, known as euchromatin. So what the cell needs is called euchromatin. That is what is on. That's what makes the cell work. That's the one that we are transcribing to give the cell its integrity and function. While the others are going to be off. These are known as the heterochromatin. And the heterochromatin, that's going to be off. So everything else is off, while the one that you need is on. So what makes the cell different is not that the DNA is different. The DNA is the same. It's which region is active. What is the euchromatin and what is the heterochromatin? Are we clear?
So how you know the genes is on or off? How you know the genes is on or off?
Yeah, so how do we know it's on and off? It's the transcription enzymes. So transcription enzymes are going to target the region in the DNA that's going to allow for the transcription of that particular gene that's going to give you the protein of interest. So there's enzymes. We're going to learn today that there's proteins and enzymes. It's called genetic expression. It's called genetic expression. This genetic expression. This genetic expression, because you know you express on and off. If I need you to give me protein, I express you on. If I don't need you, I shut you off. That is known as.
So for better understand for us, this can be applied in real life. For example, when the doctor ordered blood for patient and they told me I need troponin for this patient, or they want to do for the hemoglobin or the iron, like specific.
So the troponin is supposed to be sequestered inside the cardiac cell. So when somebody has a heart attack, the cell is going to fragment and all the stuff inside is going to leak out. So when it leaks out, it's going to be in the blood. So when you look in the blood and you find it there, that person has a heart attack. Because it's not supposed to be there. So right now, we're supposed to be in lecture. If you're walking out now, that means that there's some problem. Because, you know, something happened. So the same, that's for troponin. Troponin and also, what's the other one? Troponin and X.
The CBC, the
No, nothing. The CBC is different. Troponin and kinase, protein kinase K. Those are the two. We look for them if somebody has a heart attack. The CBC is different. The CBC is complete blood count. They take a blood sample and they inspect. How much of your blood is red blood cell? Do you have too much red blood cell? Now you're gonna get your blood too thick, you're gonna get a blood clot. Is your red blood cell too low? Now you have anemia. Is your white blood cell too high? Because you have infection. Is your albumin too? So that's when we look at the blood as a whole, complete blood count. We count all the cells. So, but I don't understand your question. What do you mean?
Like when you explained here about the check the gene to the hemoglobin.
Yeah.
Right? You go to check specifically. That's why I give an example of the blood. You know, like if it's something like that, you just check the protein, the body.
So if I want to make hemoglobin, I go inside the bone marrow and I activate the gene that's gonna be making for me the hemoglobin. So hemoglobin is a protein, and protein comes from the gene. No protein comes from nowhere. You have to be coming from the gene, from the transcription. Okay? So does that make sense? So what did I don't understand? What did you mean? The question.
I'm just trying to bring in real life all this concept. That's why, like an example, when you say about the.
You mean like if somebody has anemia, some protein is not coming out good, they go study the gene. Maybe the gene is defective, maybe some mutation happened. That's why we can't. That's like sickle cell anemia. In sickle cell anemia, something is wrong with the gene. Because when we want to transcribe the gene, there's supposed to be a sequence of nucleotides. The sequence of the nucleotide is supposed to have glutamic acid. It's supposed to have glue. Glue means glutamic acid. But due to a mutation, due to a problem, due to a mutation, is gonna change glutamic acid to Val. What does Val stand for? Valine. That's a different amino acid. So now you're gonna end up now with a different amino acid, and it's gonna destroy the red blood cell. So that shows you we need to study the gene to see what happens so we can have the correct glutamic acid and the valine. Is that clear? Then we also have to now look at the DNA and see what is it made up of. The DNA is made up of nucleic acids. So it's not made up of sugar. It's not made up of sugar. It's not made up of fat, lipid. It's not made up of protein. The proteins help. The protein enzyme are gonna help transcription, but they are not part of the DNA. And the DNA is supercoiled on itself, supercoiled and coiled, because if you spread out the DNA inside each cell, it's like a football field. So now we coil it over and over again in something known as nucleosomes. These are known as nucleosomes. These nucleosomes are like coils, so they wrap the DNA around them, and whenever we want to transcribe it, we uncoil, we relax this coil to access the DNA gene that we want to transcribe. Is that clear?
Yes. But why they say it's double helix?
So double helix, that's how it looks like. So the double helix is very important because, you see, we're gonna now look in the slides, and we're gonna see that the double helix is the way the DNA is structured. So when you compare the DNA to the RNA, you will find out that the RNA is single helix, while the DNA is double helix. So let's look here. If you now examine, you will see that the DNA is going to base pair based on nucleotides binding, where G is going to bind to C and A is going to bind to T. And A will bind to T through how many bonds? 2 hydrogen bonds, while G is going to bind to C through 3 hydrogen bonds. So let's see that in the slide some more. Yeah, we're gonna get to that. So let's begin now. So you know that the DNA is very important because many diseases are caused by genetic mutation. How many different kinds of diseases we have? Number one, we have inflammatory diseases. Inflammatory diseases, meaning there is redness, heat, pain, swelling, loss of function. Whenever you see that, that's one kind of family. All diseases in the world, they can be separated into three different families. Family number one, inflammatory disease. Family number two is cancer and tumor, uncontrollable cell division. And family number three is congenital disease, some genetic problem, and there is some malfunction, some defect. So in the embryology, the development in the embryonic life did not complete properly, so there is some congenital defect. So these congenital defects are based on some DNA problem. So now we're going to study them, like Down syndrome. We just talk about sickle cell anemia. There are many different kinds. We're going to learn about them, but these are based on some genetic problem. Is that clear?
Well, not always. Not always.
What do you mean?
Like some congenital disease, sometimes rare condition, like it's not genetic, or it's not the disease.
Yeah, it's rare genetic disease. It's always something wrong in the DNA that led to the problem.
Or not genetic. Not genetic, I mean, not came from family.
Yeah, so not all genetic disease has to come from family. You can have
Just when they.
a mutation. Okay, so the world of genetic is very complex. Some genetic problem could be because you got it from family. Some because your family don't have a problem, it just happened to you because some mutation. You know, you have to have a good understanding of the concept of mutation.
And I was taught that because my daughter, she born with congenital heart disease.
With congenital heart
It's
disease.
supposed to be malformation, but it's no genetic. It's just happening 200,000 to 1. It's a rare.
Right, so it happened, but that is in the embryology, in the development. But it's also related to DNA. How come they told you it's not DNA? It's DNA. It's always DNA.
They investigate about this condition. The only thing they find out, they call Epstein anomaly. By a man with like 85 years old, when he died, when they do autopsy, they find out he died because his people's bowel is born with this malformation, and this doctor name is Epstein. So that's why the name came. Well, they don't find out yet. No reason like why this malformation happened.
Yeah, it's always what drive the development? What makes a zygote, you know, the zygote become blastula, become morula, become gastrulation, the formation of neural tube, the formation of the organ system, the organogenesis is the genetic that does that. So always what is guiding the process of development is the genetics. So somewhere in this time event of the development, some genetic defect happened that prevented the proper formation of the valve. You know what I mean? So it's always, because the DNA is the center of the nucleus that is going to send all the signal and it's going to say continue grow. Remember we talked about the cell cycle? You enter the cell cycle to grow. Sometimes we don't enter the cell cycle because some mutation happened and the protein is not supporting the entry. Sometimes the opposite. You divide too much. You divide too much because the tumor suppressor is not there, so you end up with cancer. So this is to tell you that you have diseases like color blindness, cystic fibrosis, diabetes, hemophilia. All of that is caused by a problem in the gene. So Mendelian genetics is going to help us discern and predict the patterns. Okay. Okay, I'll make those labs for you due tonight, okay? You can go home, do them.
Are you extending diffusion?
Yeah. Mhm. Everything is due tonight. You see, everything due May 21st. May 21st at 11:59 p.m. So now let's go back to the important lecture. Okay, so the DNA, what does DNA stand for? DNA stands for deoxyribonucleic acid. So that means when we look at the structure of the DNA, there is a deoxygenated. There is a, you see, this is the nucleotide. It has a phosphate group, the yellow group. It has a phosphate group, and it has a ribose sugar. If you look at carbon number 2, where my mouse is, you see carbon number 2? Carbon number 2, does it have an oxygen? That's why we call it deoxyribose. And what is up top here? That's a nitrogenous base. How many different nitrogenous bases do we have? Adenine, guanine, cytosine, and thymine. How many nitrogenous bases do we have? 4. Okay. 4 nitrogenous bases, and you have a deoxyribose sugar, and you have a phosphate group. So now this is why we call it deoxyribonucleic acid. It has a long thread-like molecule with uniform diameter. There are 46 DNA molecules, so 46 chromosomes, 23 from mom and 23 from dad. All the cells in your body have 46 chromosomes except your sex cells. If you are a male, your sperm has 23. And if you are a female, your egg has 23. Why is that? So in the zygote, 23 from father, 23 from mother, 23 plus 23, what do you get? 46. Because if you got 46, you combine them, that's what? That's 100. That's now like an elephant. That's not a human being. So your sex cell is a half. Okay? The DNA and the other nucleic acids are polymers of nucleotides. So we said that the DNA is a macromolecule. It's not a micromolecule. And you need to know the 3 units. A sugar for DNA is deoxyribose. For RNA, it's oxyribose. It has, for RNA, it does have that, for RNA, it does have that. How is that? Is it not better? Okay. For RNA, it has, for the RNA, it has oxygen. You need to know that. Those 3 components, sugar, phosphate, like here, sugar, phosphate, and a nitrogenous base. So now let's examine one single nucleotide. The single nucleotide is going to have 1 sugar. For the DNA is deoxyribose. For RNA, it has an oxygen on it. 1 phosphate group and 1 nitrogenous base. Now you need to know your nitrogenous bases in the sense that we have 2 classes, purines and pyrimidines. Purines and pyrimidines. Purines have double ring. And pyrimidines have single ring. So those purines that have a double ring, like adenine and guanine, have double ring each. Pyrimidines have a single ring, like cytosine, thymine, and uracil. Cytosine, thymine, and uracil. Okay. Now, if we look, you see that's your purine. How many rings do purines have? They have 2 rings. 1, 2. And how many rings do pyrimidines have? Is that clear? How many rings do pyrimidines have? Swazilia. Excellent. How many rings do purines have? So usually, because pyrimidines have a longer name, people assume that they have more ring. They assume that they have more ring. Please pay attention. I'm gonna choose you. I'm gonna ask you a question. You don't know the answer. I was gonna drop your grade for the attendance. So please pay attention. It's not just showing up and daydreaming and go home and know the question. You have to pay attention to what I'm explaining. Okay? All right. So now, double helix. So that's because remember, you asked the question, Stephanie, right? Stephanie, you asked the question. There's double helix, it means that is a macromolecule or micromolecule? Is a macromolecule that is made up of micromolecules such as nucleotide. So the nucleotide micromolecule give us the DNA macromolecule. The micromolecule is nucleotide and the macromolecule is nucleic acid. Deoxyribonucleic acid. RNA ribonucleic acid. So who's bigger, nucleotide or nucleic acid? Very good. Okay, it has a double shape, and each side piece is a backbone of phosphate groups alternating with deoxyribose. There are step-like connections between the backbones and our pairs of nitrogen bases. So nitrogenous bases are united by hydrogen bonds. So a purine on one strand is always bound to a pyrimidine on the other. So we don't bind purine to purine. You bind purine to pyrimidine. You bind purine to pyrimidine. So you must know this law. Adenine binds to thymine, while cytosine binds to guanine. Very good. And adenine binds to thymine, and cytosine binds to guanine. But remember, this binding, you need to know how many bonds and what kind of bond. Excellent. These are hydrogen bonds. And between adenine and thymine, how many hydrogen bonds? 2. And between cytosine and guanine, how many hydrogen bonds? 3. So the law of the complementary base pairing. So one strand is going to determine the base sequence of the other. So if I give you one strand, it's very easy to figure out the other because you already know the law. With every purine binds a pyrimidine. C binds with G, A binds with T. This is really important here, this figure. If you look here, what do you see? You see that you have 2 strands, like 2 snakes, but they are anti-parallel. So one has a head up, tail down, the other one has a tail up, head down. It's not like both of them have this head on the same side and feet on the bottom. No. One head up, feet down, the other one foot up, head down. Okay? Now, you also need to see that between the A and the T, 2 hydrogen bond, between G and the C, 3 hydrogen bond. So these are the bonds between the 2 snakes, between the 2 strands. So the 2 strands are connected by hydrogen bond. Not inside this, so inside the strand, that's not hydrogen bond. You see, this strand itself here, this strand where my mouse is, right here. Do you see hydrogen bond here? Here. Thank you. Do you see hydrogen bond here? There is no hydrogen bond here. There is no hydrogen bond here. So that's a covalent bond. So that's why when you heat up the DNA, Thank you. When you heat up the DNA, you break these bonds. So the two strands fall apart from one another. They divorce. But each strand is not going to fall apart on itself. Their bond together is going to fall. The one in the middle is going to break, not this. So you see you have your nitrogenous base, then you have your sugar. You have your sugar in green. And what's that on yellow? What's that yellow here? What's that yellow? That's the phosphate. Excellent. That's the phosphate. What's the name of the bond? This phosphate connecting two sugars. You see? This phosphate connecting two sugars. This phosphate connecting two sugars. What's the name of this bond? It's a type of a covalent bond, but what do we name it? It is phosphodiester. Phosphodiester bond. Phosphodiester bond. Phosphodiester bond. We're going to get to that. We said that the gene is a segment of the DNA coding for the synthesis of a specific protein. And the genome is all the genes of one person. And humans have about 20,000 genes, only about 2% of the total DNA. The other 98% is non-coding DNA. So it plays a role in the chromosome structure and the regulation of the gene activity. So the double helix was discovered in the 1900s, and Nobel Prize was awarded to Watson and Crick for discovering it. Now we talked about chromatin, euchromatin and heterochromatin. What's the difference between euchromatin and heterochromatin? Rocky. So euchromatin and heterochromatin. So you know we said some part of the gene is active. We use that active part of the DNA in order to transcribe the mRNA that we will translate to give us the proteins we need. That's euchromatin. The on, remember I said on and off? The on one, the one that's active, that's the euchromatin. The one that's inactive, the one that's not working, the one that's off, the one that is not expressed, genetic expressed, and genetic shut down. The one that is shut down is known as the heterochromatin. Known as the heterochromatin. So now let's look here. Remember what I said about the coil? The coils? You see this DNA, it has linker DNA between two coils. But there's a nucleosome. Remember the nucleosome? What did we say the nucleosomes do? Yeah, they wrap the DNA around them, making it smaller. And then you fold the DNA and the nucleosomes until you end up with a chromosome. And you have 46 of those. Are we clear? Histones are proteins that are crucial for DNA packing. So histones cluster in groups of 8 molecules. So the DNA molecule winds around the cluster like thread around a spool, and the chromatin consists of thousands of repeating nucleosomes. The nucleosomes consist of core particle. You have a histone cluster with DNA that is 1.5 turns around it. And there's the linker DNA, there's a short segment of DNA connecting core particles. Nucleosomes zigzag like an accordion, and the chromatin thrown into the complex has irregular loops and coils. Chromatin and chromosome 3. So each chromosome is packed into its own region of the nucleus. That is known as the chromosome territory. It's permeated with channels allowing regulatory chemicals to have access to the genes. In a non-dividing cell, the chromatin is non-static. So changes from moment to moment according to the genetic activity. So what do we need? We're going to turn on. So that's known as genes get turned off and on through a process known as genetic expression. So as you see here, we have during the growth and development, the chromosomes are going to migrate so that the genes on different chromosomes can partner to bring the development changes in the cell. So when preparing to divide, the cell makes a copy of all nuclear DNA. That is known as DNA replication or DNA duplication. So if you want to divide, if a cell wants to divide, it cannot give the cell that is going to come out its half of its DNA. Because now we're going to end up with a cell with 23 DNA chromosomes. That's wrong. We need cell with 46. So first, we're going to double up. We're going to multiply 46 by 2. Then we're going to separate so each cell can have its own 46. That is known as replication, DNA replication. So when preparing to divide, the cell makes a copy of all nuclear DNA. Each chromosome then consists of 2 parallel filaments of identical DNA and sister chromatids. Becomes visible with the light, and the chromatids are joined at constricted centromeres, and there are kinetochores. These kinetochores are protein plaques on each side of the centromere. So you see, those are the kinetochores that the microtubules are going to attach to when we want to separate and detach. When we want to separate and detach the 2 chromosomes, the 2 sister chromatids. Now we finished with DNA. Now we're going to look at the RNA structure and the function. Now, RNA, that represents smaller molecules that resemble the DNA. It can have less than 100 or just over 10,000 bases. So there are 3 classes of RNA. So we have messenger RNA, mRNA. We have ribosomal RNA, that's rRNA. And we have transfer RNA, that's tRNA, tRNA. So one nucleotide chain, not a double helix. So the ribosome replaces deoxyribose as the sugar, and uracil is going to replace thymine as a nitrogenous base, and that's going to function mainly in the cytoplasm. So what's the gene? A gene is a segment of the DNA that carries the code for a particular protein. So the gene is an information containing segment of the DNA that codes for the production of a molecule of RNA that plays a role in synthesizing one or more proteins. So the amino acid sequence of a protein is determined by the nucleotide segment in the DNA. So what is a gene? So the genome represents all the DNA in one 23 chromosome set. So there is 3.1 billion nucleotide pairs in the human genome. 46 human chromosomes come in two sets of 23 chromosomes. So one set of 23 chromosomes came from each parent, and we discussed that. So 23 come from each parent, and then they're gonna join, they're gonna fuse together to give you the zygotes. And genomics is the study of the whole genome and how its genes and non-coding DNA are going to interact to affect the structure and function of the whole organism. So a single gene can code for many different proteins. So a gene on average has about 3,000 bases long. And all humans are at least, we are 99.99% similar. The differences we have is about 0.01. That's the only difference in DNA. So our DNA is pretty identical. So two individuals can differ by more than 3 million base pairs. So that 0.01% can come out to about 3 million base pairs. So there are combinations of single nucleotide polymorphisms. So the single nucleotide polymorphism, meaning the variation in the DNA that's going to make you different. So there is small room for change, about 0.01%. But that 0.01% gene talk is about millions of base pairs. So 0.01%, but it's still billion. So if somebody give you 0.01% of Elon Musk money, that's a lot of money. That's a lot of money. So 0.01% of the DNA is about 2.4 million base pairs. So genomic medicine is when we study the genetic hotspots. Genetic hotspots. Genetic hotspots are regions in the DNA that are hot and known for mutation, like the one in sickle cell anemia. That one is known to be having many changes, known to be having many mutations. So we study that particular region in the DNA, and we want to find out that particular region, what leads to those polymorphisms, what leads to those changes. So we study that in the genomics. It's relevant to many disorders like cancer, Alzheimer's disease, schizophrenia, obesity, AIDS, tuberculosis, and so on. So the body can make millions of different proteins. So proteomes are the proteins that we get, and we try to link them to which genes make which proteins. The genetic code is a system that enables these four nucleotides to code for amino acid sequences of all the proteins. So the minimum code to symbolize 20 amino acid is 3 nucleotides. That's very important. So now we learn that each amino acid is made up of 3 nucleotides. If you change one nucleotide, you may end up with a change. So if you will learn that each nucleotide is going to come with 2 other nucleotides, and that's going to represent something. So if you pair up 3 nucleotides, that's going to give you a particular amino acid. If you pair 3 nucleotides, that's going to give you a particular amino acid. So we will see that some amino acids are known as some nucleotides. When you pair 3 of them, they don't give you an amino acid. They give you a stop codon. So they stop the process of the translation. You will see that in just a little bit. For now, you need to know that we have 20 amino acids, and those 20 amino acids are made by a combination of 3 nucleotides. And by the way, think of a nucleotide as a number. So the nucleotides are like nucleotides. You have alanine, valine, cytosine, and glutamic acid, and you have different ones. We will talk about that now. But you refer to them, let's say, as numbers. So you have amino acid that is made by 3 nucleotides. Each nucleotide, let's refer to it as a number. So you pair up 1, 2, 3, you get a nucleotide. 1, 3, 4, you get a amino acid. 4, 5, 6, you get an amino acid. 4, 6, 7, you get another amino acid. So an amino acid is made up by how many nucleotides? 3. You're gonna learn that this code is degenerate. What does degenerate mean? Degenerate means you can end up with the same nucleotide by different nucleotides. You can end up with the same amino acid from different nucleotides. So for example, 1, 2, 3 can give us valine. If you don't have 1, 2, and 3 paired up and you had 1, 7, 8, that can still give you valine. So that means the code is degenerate in the sense that one amino acid can be coded for by different combinations of nucleotides. Is that clear? Exactly. And those 3 nucleotides will give you, like let's say you want to get amino acid, glutamic acid. You put 3 nucleotides. Then if you put the nucleotides, we know the nucleotides, adenine, guanine, cytosine, thymine. You put adenine, guanine, cytosine, you get glutamic acid. Then you put adenine, thymine again, you get still glutamic acid. So that's going to show you multiple nucleotides can give you one amino acid. That's good because if one combination is messed up, another one can be a safety and can be a substitute. We're going to see that now. So it's a base triplet. So a sequence of 3 DNA nucleotides is going to stand for one amino acid. So the codon, so the 3 nucleotides are going to make up a codon. So the 3 base sequence in the mRNA. So the codon is what we get from transcribing the DNA. Remember we said in the essential dogma of biology, we transcribe DNA in order to get mRNA. And then you have 60, and then you have 20 amino acids, and there's 61 code for amino acids, and there are 3 stop codons. So the 3 stop codons, you need to know them. You go away, you are away, you are gone. UGA, UAA, UAG. If your ribosome reads you go away, you are away, you are gone, that means it's stopped, and we're not going to continue the translation going. So if you have one of those, if you have a mutation, let's say you are supposed to have TAG. TTAG, but by a mistake, the T was removed and a U was placed there. When the ribosome is reading, it doesn't read TAG. It now reads UAG. What's going to happen now? We're going to stop the translation. That's the end message signal, like a period at the end of a sentence. Like a period at the end of the sentence, we're going to shut down this translation. And there is a one universal start codon. It's very interesting. We have 3 different stop codons, but one start codon. The start codon is AUG. AUG. You need to know that. You need to know which one is the start codon, AUG, and what are the 3 stop codons. What are the 3 stop codons? You go away, You are away, you are gone. Or you can make it only one. Okay. How many possible codons are going to represent those 20 amino acids? 64 possible codons to represent the amino acid. Very good, very good, very good, very good. Okay, all body cells except sex cells and some immune cells contain identical genes. We said that. Different genes are activated in different cells, and any given cell uses 1/3 to 2/3 of its genes. The rest remain dormant and may be functional in other types of cells. When a gene is activated, the messenger RNA is made, and the mRNA is complementary to the gene. What did we call this? Going from DNA to mRNA and from mRNA to protein. No, but the whole, yeah, that's true. From DNA to mRNA, that's transcription. From mRNA to protein, that's translation. Doubling up the DNA is known as the doubling up the DNA is known as replication. Replication, duplication. So now transcription is copying the genetic instructions from DNA to mRNA. And the RNA polymerase is an enzyme that binds to DNA and assembles mRNA. Certain DNA-based sequences are gonna signal the start. So this TATA box, TATA, this is a certain DNA-based sequence that's gonna begin for the RNA polymerase. So ACE, what's the ACE? ACE, that's the enzyme. The RNA polymerase, meaning it's gonna make RNA polymer. RNA polymerase, it's an ACE enzyme that's gonna polymerize for us RNA. So what is that gonna give us? RNA polymerase? It's gonna give us? It's an enzyme, but what is it gonna give us? What is the enzyme gonna catalyze? It's gonna catalyze mRNA. It's gonna catalyze mRNA from what? From DNA. That's why we call it DNA-dependent RNA polymerase. Because it's dependent on a DNA in order to give us RNA. Are we clear? So that RNA polymerase, in order for it to start working, what is it going to do? It's going to bind to the TATA box, and the RNA polymerase is going to open up the DNA helix, and it's going to read the base pairs from one strand of the DNA. It's going to make corresponding mRNA, where it's going to find C on the DNA, and it's going to add G to the mRNA. It's gonna, whenever it finds G on the DNA, it's gonna add C on the mRNA. So it's matching. It's reading one strand and creating an mRNA strand based off of it. But remember, there is something very important, and that is when it finds an A, when it finds an A, what is it gonna make? A U. Usually, the DNA polymerase, what's the difference between an RNA polymerase and a DNA polymerase? What's the difference between an RNA polymerase and a DNA polymerase? You're talking about DNA and RNA. I'm talking about DNA polymerase and RNA polymerase. So those are two enzymes. DNA polymerase is going to make for us DNA. So we're going to use that in the replication. want to double up the DNA, we use DNA polymerase to double up the DNA. When we want to make RNA, we use RNA polymerase to make RNA. The DNA polymerase, when it sees A, is going to give us, no, the DNA polymerase. This is the RNA polymerase. The RNA polymerase making RNA, when it sees an A, is going to put a U. mRNA doesn't have a T. But the DNA polymerase has a T. So it finds the A is going to put a T. So that's the DNA polymerase, RNA polymerase. The RNA polymerase rewinds the DNA helix behind it when we are done, when we are closing down. And then that RNA we make, what are we going to call that? We're going to call that the nascent mRNA transcript. The nascent mRNA transcript is an immature RNA produced by transcription. Now, there are regions in the mRNA that must be cut out and regions that are kept. So, region A, we want that. Region B, we want that. Region C, we want it. D, we want it. E, we want it. F, we want it. The ones in the middle, those are like need to be removed away. So you see what you have up here. That's the DNA. When you transcribe the DNA, now you have mRNA is nascent, meaning that it's not ready because it has some intron. So the introns have to be excised and cut out. The exons are gonna be what we need. So we're gonna keep the exons, or we're gonna remove the intron. We don't want the intron. That's why the introns get to stay in the nucleus. They're not gonna go to the cruise to the. They're not gonna go to the ribosome to be translated. Are we clear? They're not gonna go to the ribosome to be translated. So you see here, that's why we call them intron, because they're in the nucleus. Where is the transcription happening? Tomorrow, where is transcription happening in the cell? Where, not when, where, a place. Where is it happening? In the nucleus. It's happening in the nucleus. The transcription is happening in the nucleus. And the translation is happening where?
Outside.
Where? In the cytoplasm. So that means if you want to be translated, you have to go outside. Exported. That's why the exon is going to exit and go for ribosome translation. But the intron is going to stay behind. In.
The intron is part to the transcription. The intron is part of the transcription, and the exon is part of the.
Both of them are part of transcription. You see transcription? Transcription gave us both. Gave us exon and intron. But we're going to do some post-transcriptional modification. We're going to do surgery. We're going to do modification, remove what we don't need, and keep what we like. Is that clear? And then the translation, we're going to translate the mRNA to get our protein. Are we clear? The RNA polymerase is going to rewind the DNA helix behind it, and the gene can be transcribed by several polymerase molecules. And the terminator is going to be a base sequence at the end of a gene that's going to signal a stop. Pre-mRNA is immature RNA produced by transcription. Exons. What are exons? You see where my mouse is? The exons are segments of the pre-mRNA that will be exported. Exon exported out. That's what we need. But introns are gonna stay in. But you know they are not useless. They're gonna be recycled, and it's called alternative splicing. This alternative splicing is going to cause the alternative splicing is going to recycle all the introns and make something useful out of them. Are we clear? So in alternative splicing, there are variations in the way exons are spliced, and they allow for a variety of proteins to be produced from one gene. So one gene can code for more than one protein. So exons can be spliced together into a variety of different mRNAs. Translation is a process that converts the language of nucleotides into the language of amino acids. There are three main participants in translation. So mRNA is going to carry the code from the nucleus to the cytoplasm. It has a protein cap that is a recognition site for the ribosome. So now we moved from transcription into translation. And there is a transfer RNA. So now we talked about mRNA. We're done with mRNA, which is a transcript. You know, you go to the pharmacy, you give them a prescription, they can read it, give you the medicine. No two people can have the same transcription. You have to have your own transcription. But that's not the only RNA we have. We have also tRNA and rRNA. rRNA is ribosomal, so that one is part of the making the machinery of the ribosome to be able to translate DNA. Is that clear? Now, transfer RNA is gonna, from the name, I wish that instead of calling it transfer RNA, that they called it translation RNA. Because that's what it does. It just makes sense. But no, they have to confuse it. But the translation RNA or the trans is not translation RNA. If they ask you an example, the T stands for, you put translation in your home. But that's what it does actually. They call it transfer. Why? Because in the translation, it transferred the amino acid. It's going to transfer the amino acid from the peptide into the polypeptide sequence that we're going to be generating. So we're going to look at a video of how the ribosome is going to work. So in the translation, you have the mRNA is going to carry a code from the nucleus to the cytoplasm. We know that. That's what we're going to be reading. It has a protein cap that is recognized, a recognition site for the ribosome. And the transfer RNA is going to deliver a single amino acid to the ribosome for it to be added to a growing protein chain. Then you're going to have the ribosome, and these are going to be organelles that read the message. So the mRNA is going to have the leader sequence, and it's going to act as a binding site for small ribosomal units. And it's going to be there's going to be a large subunit and a small subunit. For eukaryotes, it's a 40-60 subunit. For prokaryotes, it's 30-50 subunit. So let's look here. What did we say the start codon is? The start codon for translation is AUG. And when we read AUG, we're going to be able to start the process of translation. And when we read, you go away, you are gone, you are away, what are we going to end up with? We're going to end up with a stop coda. We got to stop that. Another thing you need to know is that the start codon, it will start translation and give you what amino acid. But the stop codon, it will stop but will not give you anything. So the stop codon is just to stop the ribosome from making a polypeptide. But the start codon, AUG, will give you an amino acid. What's the name of the amino acid the start codon will give you? Methionine. This methionine is the amino acid that's going to be given by the AUG start codon. tRNA, transfer. You see, not translate. It's a small RNA molecule. It coils on itself to form an angular L shape. One end includes 3 nucleotides called an anticodon, and the other end has binding sites specific for 1 amino acid. The tRNA is going to pick up 3 amino acids in a cytosol, and the cost of binding an amino acid to the tRNA is 1 ATP. That's very important. So we need 1 ATP in order to hook up and attach the amino acid to the ATP. Who's that? Who's that? That's the transfer RNA. Like a hairpin. You know the hairpin? Yeah, it looks like a hair. It carries on one side the anticodon. And on its tail, what does it have? That's the amino acid accepting site. We will see in a little bit. So when we begin translation is comprised of 3 steps. You have initiation, you have elongation, and you have termination. Initiation, elongation, and termination. So initiation represents a leader sequence in the mRNA is going to bind to small ribosomal subunit, and the initiator tRNA is going to be bearing methionine, is going to pair with the start codon. The large ribosomal subunit is going to join the complex, and the now fully formed ribosome is going to begin reading the bases. Elongation. So next to the tRNA with its amino acid is going to bind to the ribosome, while its anticodon is going to pair with the next codon of the mRNA. The peptide bond is going to form between methionine and the second amino acid. The ribosome is going to slide to read the next codon and release its initiator tRNA. So that's the second step after initiation. You're going to have elongation. The process continually repeats, extending the peptide to a protein. And in termination, when the ribosome reaches the stop codon, a release factor is going to bind to it. And when we finish, the protein is going to break, the finished protein is going to break away from the ribosome, and the ribosome is going to dissociate into subunits. So the translation can be rapid, and the cell may have about 300,000 identical mRNA molecules. Let's now look at a video of how translation happens. It's very important to look at a video of how translation happens. Ribosomal translation.
Initiation. The small ribosomal subunit binds to the initiator tRNA carrying the initiator amino acid methionine. This complex then attaches to the cap structure at the 5' end of an mRNA and scans for the start codon AUG. The process is mediated by several initiation factors. At the start codon, the large ribosomal subunit joins the complex and all initiation factors are released. The ribosome has 3 sites. The A site is the entry site for new tRNA charged with amino acid or amino acid tRNA. The P site is occupied by peptidyl tRNA, the tRNA that carries the growing polypeptide chain. The E site is the exit site for the tRNA after it is done delivering the amino acid. The initiator tRNA is positioned in the P site. Elongation. A new tRNA carrying an amino acid enters the A site of the ribosome. On the ribosome, the anticodon of the incoming tRNA is matched against the mRNA codon positioned in the A site. During this proofreading, tRNA with incorrect anticodons are rejected and replaced by new tRNA that are again checked. When the right amino acid tRNA enters the A site, a peptide bond is made between the 2 now adjacent amino acids. As the peptide bond is formed, the tRNA in the P site releases the amino acids onto the tRNA in the A site and becomes empty. At the same time, the ribosome moves 1 triplet forward on the mRNA. As a result, the empty tRNA is now in the E site, and the peptidyl tRNA is in the P site. The A site is now unoccupied and is ready to accept a new tRNA. The cycle is repeated for each codon of the mRNA.
You see how it's made?
Termination.
So the ribosome has 3 doors: EPA, exit, and the P site is the peptide, where the peptide chain is multiple amino acids. Remember we said the protein primary sequence is a polypeptide, then it folds upon itself to make a secondary peptide, then to fold upon itself to make a tertiary structure, then fold upon itself with some more disulfide bridges, ionic bonding to make a quaternary structure. So that's the peptide in the middle. Then the new one is going to be coming at the A site. So the ribosome is going to be shifting towards the A site, taking the peptide, shifting it over 1 step, and the empty tRNA is going to be thrown out. This is very important to understand how it works.
Termination happens when one of the 3 stop codons is positioned in the A site. No tRNA can fit in the A site at that point.
You are gone.
tRNA can match that sequence. Instead, these codons are recognized by a protein, a release factor. Binding of the release factor catalyzes the cleavage of the bonds between the polypeptide and tRNA. The polypeptide is released from the ribosome. The ribosome is disassociated into subunits and is ready for a new round of translation. AUG. tRNA, the tRNA that carries the growing polypeptide chain. The E site is the exit site for the tRNA after it is done delivering the amino acid. The initiator tRNA is positioned in the P site. Elongation. A new tRNA carrying an amino acid enters the A site of the ribosome. On the ribosome, the anticodon of the incoming tRNA is matched against the mRNA codon positioned in the A site. During this proofreading, tRNA with incorrect anticodons are rejected and replaced by new tRNA that are again checked. When the right amino acid tRNA enters the A site, a peptide bond is made between the two now adjacent amino acids. As the peptide bond is formed, the tRNA in the P site releases the amino acids onto the tRNA in the A site and becomes empty. At the same time, the ribosome moves one triplet forward on the mRNA. As a result, the empty tRNA is now in the E site, and the peptidyl tRNA is in the P site. The A site is now unoccupied and is ready to accept the new tRNA. The cycle is repeated for each codon of the mRNA.
Okay, so you can watch this some more at your own time later. Okay. Okay, you can just type on the translation process on YouTube, and you should be able to find many different animations that are going to help you a lot. Okay, that shows you the process. You see, that's the E site. You see the E site, the P site, and the A site. And you know, we're going to be shifting the peptide sequence from the P site to the A site until we read a stop codon. Then we terminate everything, and the exit site is the E for exit. P is for peptide formation, and the A site is the site of the entry of the new tRNA. So after translation, some proteins are packaged and some others are exported. And proteins headed for the lysosomes for secretion are made on ribosomes. Okay, so now you need to know this very important fact. We said that, remember earlier when I was drawing the ribosome, I said there are free-floating ribosomes in the cytoplasm, and there are rough endoplasmic reticulum bound ribosomes. Remember that? You remember that? So now it's very important to look at the significance. Why is that? Why do we have a subset of ribosomes that are free-floating in the cytosol? And we have a subset of ribosomes that are embedded on the endoplasmic reticulum. That's why we call it a rough endoplasmic reticulum. Why is it rough? Because it has ribosome on it. So what's the difference?
Because they make proteins in the DNA.
And in the free-floating, they don't make proteins. It's a ribosome. What does the ribosome do? Make proteins. So whether you are in cytoplasm, whether you are in endoplasmic reticulum, you will make proteins. The difference is what kind of protein you're going to be making. Okay. So the proteins that are going to be made in a free-floating ribosome in a cytoplasm, they're going to be used by the cell itself, locally, inside the cell. And the proteins that are going to be made by the rough endoplasmic reticulum, they're going to be exported out. They're going to be sent out of the cell. So one is Amazon, it's going to be shipped all over, and the other one is going to be used inside the cell. So protein processing and secretion. So the protein synthesis is not finished when the amino acid sequence or the primary structure has been assembled. So in order to work, the protein must fold into precise secondary and tertiary structures. There are chaperone proteins. So the chaperone proteins are all the proteins that pick up new proteins and guide their folding into the proper shape. And they help prevent improper association between different proteins. So some also called stress proteins or heat shock proteins. So they help damaged proteins fold back into the correct functional shape. So protein processing and secretion. You see, now you see the proof. This is the endoplasmic reticulum. It has the ribosomes around it. So now they're gonna send their proteins into the Golgi complex. See this pita bread? You have many pita bread on top of each other. That's your Golgi body. You see what the Golgi body does? It's gonna send you out. So all of you see protein formed by ribosomes on the rough endoplasmic reticulum. The protein is going to be packaged into a transport vesicle. The transport vesicle is going to fuse into a cluster that's going to unload the protein into the Golgi body. Then the Golgi body is going to modify the protein structure, and the Golgi vesicle containing this finished protein is going to be formed, and the secretory vesicle are going to be released. They're going to release the protein by this exocytosis. Are we clear? Proteins to be used in the cytosol are likely to be made on free ribosomes in the cytosol. So proteins destined for packaging into the lysosomes or secretion from the cell are assembled on rough endoplasmic reticulum, and they are sent to the Golgi complex for packaging. So the entire polyribosome is migrated to the rough endoplasmic reticulum and is going to dock on its surface. So the assembled amino acid chain is going to be completed on the rough endoplasmic reticulum and then sent to the Golgi body for final modification. Then that protein is going to be processed and sent out. So the genes, we talked about the gene regulation. We can turn the gene on and then off. The cells can turn some genes permanently off. And example would be liver cells turn off hemoglobin gene. Liver cells don't make hemoglobin. So the hemoglobin gene is going to be off in the liver. But liver cells make albumin. So albumin is always going to be on in the liver. That's why people with liver cirrhosis, they don't make albumin. So they always have a problem. Alcoholic liver disease, cirrhotic liver disease. These patients, they cannot make albumin, so the osmolarity, the osmotic balance, there is no U osmolarity in their blood. The tissue has more proteins than in the blood, so the water is constantly leaking out from the blood into the tissue compartment, making tissue edema because there's no protein, the albumin. So that's an example now showing you gene regulation. So casein is a milk hormone. So the hormone prolactin is going to bind to receptors on the membrane of the mammary cell. Receptors are going to trigger the activation of this transcription activator in the cytoplasm. The regulatory protein is going to move into the nucleus and bind to the DNA near the casein gene. The binding enables RNA polymerase to bind the casein gene, transcribing it, giving you casein mRNA. Then the casein mRNA is going to go to the cytoplasm and ribosomes on the rough endoplasmic reticulum. So that means we're going out. It's going to be exported out. The Golgi complex is going to package it, and a secretory vesicle release the casein by exocytosis it becomes part of milk. Look at it here. So prolactin is gonna bind the prolactin receptor. So the signal is gonna bind the receptor. And then we're gonna hydrolyze ATP to ADP, and you're gonna have a regulatory protein known as transcription activator. This transcription activator is gonna go and it's gonna find the casein gene. You see the casein gene? It's gonna transcribe the casein gene to give you see the RNA polymerase is gonna transcribe the casein gene, is gonna give you what kind of mRNA? Casein mRNA. Because we transcribe casein gene, we're gonna get casein mRNA. Then we're gonna go to the rough endoplasmic reticulum bound ribosomes. They're gonna translate for us the mRNA for casein to give us the actual casein into these vesicles. Then we're gonna send it into the Golgi body. The Golgi body is gonna package it into vesicles. Then we're gonna exocytose casein, which is gonna be part of milk. And it's very important for the baby. So that shows you what happens. And our signal caused the transcription of the casein gene. And then we got an mRNA for casein. It was translated in the rough, not the free-floating ribosome. The rough endoplasmic reticulum bound ribosome that's gonna call the gold. It's gonna be bound to the Golgi, and then it was sent out. Are we clear? Okay. We talked about the gene replication. We said that genetic replication is semi-conservative. What does semi-conservative mean? First of all, we need to unwind the double helix. If we want to double the DNA, we need to unwind the double helix and double up each strand by itself. So you see, that was a double strand. We separated it into two strands, then we doubled each strand. So we began with two parent strands, and we ended up with two daughter strands. That's called semi-conservative. It's not 100% conservative, but we don't end up with two parent. We began with two parent. These are the two parent, 1, 2. But from each parent, we doubled up a daughter. From this parent, so you see, we were double here. Do you see that? Parental DNA, double. We separated it into two parent, and each parent was double on itself. That's why we call it semi-conservative. You need to know this DNA helicase, that's the one that's gonna unzip and unwind the DNA. Unwind the helix, for helicase. You need to know that the DNA polymerase is going to be working from 5' to 3'. But since the two DNAs are anti-parallel, one is 5' to 3', one is 5' to 3', one of the two sides is going to be lagging. One of the two sides is going to be making fragments that must be glued up, ligated with glue. That's the enzyme known as DNA ligase. Enzyme known as DNA ligase. So the DNA is semi-conservative and discontinuous. Discontinuous because as you see here, some parts need the ligase, the glue. These are known as the Okazaki, after the Japanese scientists who discovered them. What is the Okazaki fragments? Okazaki fragments. The DNA polymerase does make mistakes, but very rarely. When you have mutations, these are changes in the DNA structure due to the replication. Okay, that's the cell cycle. We said that the cell cycle has two phases. This whole green phase is known as interphase. It's made up of three phases. G1, that's the first gap phase. It has growth, normal metabolic roles. DNA replication, growth, and preparation for mitosis. There's interphase, and all these little steps, these are known as the mitotic phases. So G1 is the first gap phase. S is where we double the DNA synthesis. And G2 phase is the second gap phase. In prophase, you find condensation of the nuclear envelope is going to break. So you have to go and find which segments are distinctive, what features, what significant features happen during each phase. In prophase, the chromosomes condense and the nuclear envelope is going to break down. The spindle fibers are going to grow from the centrioles, and the centrioles are going to migrate to opposite poles of the cell. The metaphase, you're going to have chromosomes that are going to lie along the midline of the cell, and some spindle fibers are going to attach to the kinetochores. Fibers of the aster are going to attach to the plasma membrane. Then in anaphase, you're going to have the separation, the plucking off of the chromosomes. And in telophase, you're going to have the division of the cytoplasm. You're going to end up with 2 daughter cells. In cytokinesis, there's a division of the cytoplasm into the 2 cells. And in telophase is the end of the nuclear division. Okay. So we said that there are 22 pairs, each one, 22 pairs, each one is 23. So that comes up to 46 chromosomes. But one pair of sex chromosomes, each is 23. So that's an X and a Y. So you know haploid meaning you have N, so that's half the number, and diploid is 2N, so that's 46. Diploid is 46 and haploid is N. Is a dominant allele, that's the allele that's gonna ensure that a dominant trait is gonna be passed, while the recessive allele is the allele that's gonna represent the weaker trait. Okay, so that shows you here, we pair up, if you have 2 homozygous dominant, you end up with a dominant trait. And if you have 2 homozygous recessive, you end up with a uncleft chin. What is better, cleft chin or uncleft chin? Uncleft? You think cleft chin is better? What is better? Uncleft. Some people used to think cleft chin is better. Huh? It depends? Let's see. Okay. Okay, so yeah, I think that's all. Let's go and do the lab now. Let's move to our lab. Let's do the virtual labs, and then we